You all today. We have first to thank our sponsors, the Rutgers School of Arts and Sciences, the Rutgers Science Explorer, the Institute of Earth, Ocean, and Atmospheric Sciences, and of course, our friends of the Rutgers Geology Museum. We have an interesting lineup of programs for you today. We have lectures starting at 10 o'clock in just a few minutes, 11:30 and 1. We have children's activities going on at 9:30, 10:30, and 1. And our virtual mineral sale is now live on Facebook. The QR code there can be scanned for more information, or you can visit our website at rutgersgeologymuseum.rutgers.edu. This is one of many virtual programs we've been doing during the lockdown. They include mineral patch programs, geology merit badge program events, virtual paint-alongs, our very popular Ask a Geologist series, and late night events. So there's more information on our website or with the QR code that you see on your screen, and we hope that you'll participate in other events. If you've enjoyed these and you'd like to become one of the Friends of the Rutgers Geology Museum, I'd invite you to become a member. We have different levels of membership. Benefits include free admission to members-only events, museum t-shirts, and more. So please, if you can, support the museum by becoming one of our friends. And now I'd like to introduce our first speaker. Dr. Sarah Sheffield is an evolutionary paleobiologist. She studies the fossil record to see how organisms have changed over time. Her research specializes in the fields of evolutionary paleobiology and invertebrate paleontology. Dr. Sheffield earned her bachelor's degree in geological sciences from the University of North Carolina at Chapel Hill. She went on to complete her master's degree in geology at Auburn University and then received her PhD at the University of Tennessee. Currently, she's an assistant professor at the University of South Florida, where she teaches classes in general geology, sedimentology, and paleontology. And today, Dr. Sheffield is going to speak to us about unraveling the evolutionary relationships of early echinoderms. So, Dr. Sheffield?
Thank you so much for inviting me. I'm so excited to be here. I'm going to go ahead and share my screen with you. So, as Kathleen so kindly, gracious, one moment, we just practiced this. All right, here we go. So thank you again for having me here. I'm so glad to be here, so excited to be talking to you about science. It is so nice to be able to talk to some people about science. I feel like I've been talking to my dogs a lot lately since I've been, working from home, and they just don't care that much about paleontology, I gotta say. So glad to be here. So today I'm going to talk to you a little bit about unraveling the evolutionary relationships of early echinoderms. So to do this, we're going to be spending a lot of time in the geologic record today. And this is just an artful description of what that looks like. So here we are up here with the pickup truck and the cute little dogs. But today we're going to be traveling really far back in time. We're going to be going past the mastodons, past the dinosaurs. I know, past the dinosaurs, but don't worry, I'm selling you on something much cooler than dinosaurs. All right, and all the way back to a time that looks really different from what we have today. And we're going to be talking a lot about echinoderms. So our research goals for today, or what we're going to learn about today, is we're going to be finding new fossils in unexplored areas. We're going to be looking globally in new areas, and we're going to be looking for new preservational features that can give us more information about these echinoderms. And we're going to be using these fossils either from fieldwork, finding new ones, or museums to help us better put together what we know about the evolution of these groups. So just in case echinoderms weren't on the forefront of your brain today, we're going to define them really quickly before we move on. Echinoderms include the friendly groups we all know and love, like the sea stars, brittle stars, the crinoids, otherwise known as sea lilies, the sea urchins, and the cucumbers. Those sea cucumbers, those are the 5 groups we have today. And they're defined by something we call five-ishness. And that's the actual term, I promise you. That's what scientists use, five-ishness, right? And we can see that with the starfish, 1, 2, 3, 4, 5. So they have a calcite skeleton. pretty much like limestone. So what the entirety of Florida is made of. And then finally, they have a water vascular system. So if you've ever seen a sea urchin or a starfish move a little bit, that's their water vascular system. It moves water to different areas of the body, which allows them to feed, move, and do a whole bunch of other cool things. But that isn't all there is. In the fossil record, we actually see a huge diversity of echinoderms we don't see today. So again, we're going to look at this beautiful photo here, and we're going to be going all the way back to the Paleozoic. And specifically, we're going to be camping out in what we call the Ordovician, which is about 488 million years ago. So a really long time ago. And this is the type of thing we're going to be looking at. This is beautiful. This is an early example of a starfish fossil. Absolutely gorgeous. This name is Astropecten. Beautiful. Now, one of the things I want to point out here is, do you see all these little pieces? Each one of those is an individual part, just like humans have 206 to 210 bones in their body for an average human. This has a lot of pieces to its body, and I'm going to ask you how many you think it has in just a second. And here's another example. This is one that might not be so familiar to you, but it is also an echinoderm. This is one of my very favorite fossils, Eumorphocystus, and that's from Oklahoma. So these photos are a little bit misleading. And the reason for that is these are so beautifully preserved that we can see so many of the details on its body. And as I said, echinoderms have a lot of pieces. So this usually works a little bit better when I'm talking to humans face to face, but I'm going to pretend like I'm going to ask you a question. You're going to shout out things back to me. So I'll just pick some random guesses here. How many pieces to an echinoderm body do you think there are? Okay, so I'm going to go ahead and give you the answer because I imagine now you're shouting at the screen. I can't hear you. So that's not going to work too well. But there are over 1 million pieces to the average starfish. So you can imagine that when this starfish dies, it can fall apart like that, which means most of our fossils don't look that beautiful. I mean, they look beautiful, just maybe not like that. So here's how this works. After death, here we have a bunch of different, like this is how they look right after death. So within an hour, usually these things will actually start to break up. Within 2 weeks, they're almost unidentifiable. They're pretty much just sand at that point. So fossils are incredibly rare. So when we find one, it's a gift from the earth, right? So let's take a look at what these are going to look like. The fossils that we're going to be talking about today are in this group here, which means that most of them are going to be pretty much done for within about 2 weeks. And this is often what they look like. This here on the left. This is a Holocystites, that's its scientific name, from Wisconsin. And here we have another one, this is from Indiana, called Polycystis. And even though this one looks beautiful, it's actually still missing a lot of pieces to its body. It has cover plates over these two holes. This is the mouth, this is the anus. It's missing its arms, it's missing a whole bunch of its body. So one of the things that we can do is, even though we can look at places that have been looked over before with fossils, if we go to places, as an example, Indiana is a famous fossil collection site for these types of fossils. These are echinoderms. You can see that little starfish shape, 1, 2, 3, 4, 5, that nice little five-ishness there. We can actually find new preservational features. These two fossils are absolutely magnificent in preservation. These preserve extremely rare pieces, these plates covering the hole, this is the mouth, and then these plates covering the hole, this is the anus. And then here's another one that's actually an arm preserved, so that would come off the top of its body, and this would be how it would collect food. So there'd be like little, kind of like your nose hairs, cilia, you know what I mean, like little sticky hairs, that kind of would line down this way and kind of move down and feed towards the mouth, if I were an echinoderm. So one issue we have with understanding more about echinoderms is what we're going to call a geographic bias. Now, you're going to see a chart like this a whole bunch today, and I'll try to remind you every time we look at it, because each one can look a little bit different. But this is the world about 480 million years ago. So on a land called Laurentia, this is where most of the United States was. So this is about where New Jersey was 480 million years ago. It was a tropical paradise. I don't imagine you were thinking that that was such a good move for plate tectonics to move New Jersey to where it is in its current day. I understand you're about to get a big snowstorm. But this is where most of the United States is. This is where parts of Russia are. This is where Norway, Sweden is. This is where the UK is. And this is where South America, Africa, Australia, and Antarctica are in a giant continent called Gondwana. So this is what the Earth looked like a bunch of years ago. And unfortunately, in the modern day world, most of what we know about echinoderms and about this time period come from Laurentia. So scientists have primarily studied this part of the world while leaving this part of it blank, which means we're really missing a lot of data, which is a problem because more data can tell us more about this beautiful story we're learning. So even one fossil can totally change what we're looking at. So this is Sinus. It's also an echinoderm. It does have that five-ishness, even though it looks a little squashed in this picture. This is a single fossil from about 520 million years ago from China that completely changed what we thought we knew about the evolution of echinoderms. Here's another one. This is the Ordovician to Silurian boundary. Those are two words that say time units, essentially. So it's 443 million years ago. Here we are in the Anacostia Island, which is a fairly not so easy place to do field work in because it's pretty remote. But here are a bunch of lovely paleontologists looking for echinoderm fossils in the middle of a river. And this is what they found. This beautiful specimen right up here. Again, we see that five-ishness, 1, 2, 3, 4, 5, indicating it's closely related to our good old friend, the starfish, right? This is the earliest known fossil of its kind from North America by about 15 million years. And that's a really big jump in what we knew. So looking at new places and finding new preservational features, so things that preserve slightly different things than we're used to seeing, can really help us understand more about the evolutionary relationships of our fossils. So I'm going to tell you one more quick story. Here we go to Sardinia, Italy. This map is in Italian, just heads up if you're trying to read it. And Sardinia, Italy has not been well explored for a lot of fossils, especially echinoderms. I don't know why. It's a beautiful place to do field work, I gotta say. But so over here, we have a lot of the rocks of the right age that I was talking about. So about 440 million years ago, the Ordovician age. And here is a group of scientists doing field work with me. Here I am in the back with a number of other colleagues doing field work. And within 1 week, we actually found a ton of new echinoderm species. Here they are. Here is one of our assistants breaking open the rocks to find these fossils. This is what the other side of the field work looked at. I'm telling you, you should really go here and do some field work. Absolutely beautiful, right? So this is what we found in 1 week of field work. 12 new species of echinoderms, which is a lot of new diversity. And this actually helps us fill in a lot of information about what we thought we were missing. And we'll talk more about what kinds of things that we were missing. So here are more of those echinoderms that we found in Sardinia. Absolutely beautiful. All of these are quite small. I know when I project them on the screen, they look quite large, but the largest of them is probably going to be about that big. So you can see the scale bars, but sometimes it's a little hard to imagine. All right, so we're going to take a look at a lot of trees today, specifically phylogenetic trees, not plant trees. So how we're going to read this is we're going to read this like a family tree. So think of it like the ancestors, like your great, 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 great times about a bazillion grandparents here at the bottom. And the ancestors are going to be at the top. These lines here, the more closely they are connected, that means the closer relationship they are. So these two here that share this, we can think of them as like sisters, and that's actually what we call them, sister groups. So you don't need to worry about how to read every single line on this, but that's the basic breakdown of it. So here is the evolutionary tree of Echinodermata. This is one hypothesis. We are going to continuously test this, and we're going to change some things, but there are thousands of species in this tree, right? Because we have a bunch of different species of starfish and things like that. And one of the things that we can see is that most of our living stuff, the modern stuff, is kind of clumped over here. We have one group here, the crinoids, that are kind of hanging out with some of our extinct friends, meaning no longer living. And we're going to take a look at understanding how this tree is made and what questions we still have about it. So one question I have is echinoderms evolve super, super high diversity. And they do it really fast, geologically fast, which is really like millions of years ago. But to the Earth, which is extremely old, it's kind of like the blink of an eye, right? So echinoderms evolve super high diversity. You don't need to know anything about this to say, wow, these critters look different. Let's compare this to vertebrates. Pretty much all like you and me, right? Pretty much all the vertebrates we know have 2 arms, 2 legs, and a head and a torso, right? Vertebrates are pretty boring. Even those that have made some modifications, like snakes, still have those limbs that are left over. I don't know if you knew that, but snakes do actually have a little bit of legs on them. Horrifying, right? So here are some echinoderms that look really different. We have helicoplacoids, which frankly look more like soft serve ice cream cones to me. We have eocrinoids, which have these squiggly little arms sticking up. We have all kinds of different diversity. Now, why are they evolving this different and this fast? That's a really good question, and we're going to talk about that question. Now, this graph may look a little confusing at first, but all this is saying is that the further apart these dots are, the more different they are, okay? So vertebrates, in comparison, would not be spread out across the chart. They'd be much more closely related or much more closely put together. So these are showing a lot of differences in the overall bodies. And I put some pictures here to kind of help us because these words don't mean anything to us. But as you can see, they're plotting all over the place. We have stemmed things over here. We have things that are circular over here, more circular radial. We have things, again, like this weird soft serve ice cream cone kind of hanging out in the middle. So the further the distances, the more different these echinoderms are. So today we're going to be answering, why do we think that they evolved all this different diversity? What was the point? My hypothesis is climate change driven diversity. Now, this line is something that we're going to call a change in carbon-13. And we're just going to drop the phrase carbon-13 and we're just going to talk about the amount of photosynthesis. So the amount of essentially plants and bacteria, the amount of oxygen that they're producing based on taking in sunlight and carbon dioxide. The more photosynthesis, the more oxygen that's released, so the overall cooler the environment is. So the only thing you need to know about this line here is that it is going up and down, and it's going up and down by a lot, right? We see that it goes really high here, and then it goes really low there. So we see that the temperature is fluctuating rapidly during the Ordovician. And as you can imagine, this might be really annoying to things that are alive during this time. As you can imagine, like today, we see climate change is causing a lot of issues for our flora and fauna. So it was no different in the past. So what do you think? Maybe echinoderms are evolving all this diversity because of all of the instability in the climate. Well, that's a really good hypothesis, but we need to test it, right? So how are we going to do that? So let's take another look. Now, you've looked at this chart before, just a little bit different. This is what the continents looked like 400 some odd million years ago. Again, this is where most of the United States, Canada, and Mexico are, with some exceptions. For example, Florida is still a twinkle in the geologic eye. Florida will not be around for another couple 100 million years. It's the geologic baby of the United States. And here is where Australia, Antarctica, South America, and Africa are down here. So what we see is about 470 million years ago, it gets really darn warm, super warm. So echinoderms, it's a little too warm for them at the equator. So a lot of them actually show that they get more diverse down near the South Pole where it's cooler. Well, at 450 million years, it actually gets really cold. We have a massive ice age. So echinoderms actually start moving back up towards the equator. So we do see echinoderms kind of moving through time and space, right? They pack up their bags, not literally, obviously, and they move to different places. But one of the issues is, for me to understand how they're moving, like with specific, like this thing's moving this way, this thing's moving this way, we need to understand how they're related to one another. And we actually have a lot of questions about why that is. How were they related and why are they moving? So today we're going to learn a little bit about how paleontologists make these evolutionary relationship trees that we've looked at already. So work with me. There's going to be a little bit of vocabulary, but I promise I'll explain it, and I'll use a lot of pictures to help us remind this stuff. So many traditional groups of echinoderms are what we're going to call not monophyletic, meaning an ancestor and all of its descendants. To explain what that concept means, though, I'm going to use little pictures here, okay? So there's going to be two other words, paraphyletic, meaning missing some, and polyphyletic, meaning many. So mono means one, poly means many, para means missing some. So if I were to talk about fish, I technically have to include humans. And the reason for that is that humans come from a long line of evolutionary groups that began at some point with fish, right? The first fish that crawled out of the water, we know that as Tiktaalik, the flat-headed fish that could move and push sediment out of its way on land. So technically, we're fish. And you can read a little bit more about that if you want to know more. There's a great book out there called Your Inner Fish by Neil Shubin. But if I leave humans out of this, it kind of messes with our evolutionary analysis, right? I'm removing data. Now, I'm not suggesting that you walk up to your friends the next time you see them in person, safely, of course, social distance and masks included, and say, hey, fish, how you doing? It's not going to win you friends. I've tried with my students. It does not work. But for evolutionary analysis, it really helps us to figure out who's related to who by having the whole group. And in contrast to that, we have something called polyphyletic, which means many different things are shoved together in a single group. So for example, a long time ago, scientists thought hippos and elephants were closely related. Makes sense, right? Hippos and elephants are both gray, they're both big, they're both super weird looking. Why evolution? You know, all these questions. But it turns out they're not actually closely related. They both got big and weird looking and gray for two totally separate reasons. So by combining them into one group, it actually makes our evolutionary analysis look really weird, okay? So we're going to use some more familiar examples too to help us continue to understand that. So the most important thing is, without knowing our monophyletic groups, like this, including humans and fish, we can't know anything about our evolutionary trends. So that's what we're going to be doing today. All right, so we're going to use homology to quantify or to understand our evolutionary relationships. Now, for this slide here, I will need you to pull out your human arm here. I am assuming if you're listening to this, you've got one of those. All right, so what we see in vertebrates, from the very first fish that walked on land, something like Tiktaalik, what we see is a pattern called one bone. So your humerus is that one bone, two bones, your radius and ulna, and then many bones. So your wrist and your finger bones. All right, so if we take a look at a bird, we have that one bone, two bone, many bones. If we look at a horse, one bone, two bones, many bones. A whale even has it, and that's because whales are derived from land-dwelling organisms as well. One bone, two bone, many bones. Even though it looks a little different, all of the vertebrates have this. That's something that's been descended through time through our ancestors. That's something we're going to call homologous, coming from the same place. Homo means same, all right? So what we do with evolution is we take our similarities to see how we are related to one another, all right? So let's look at some dinosaurs real quick. I know I told you we're going to talk about something cooler than dinosaurs, but dinosaurs are also pretty cool. So birds have wings, right? We also know that pterodactyls have wings. Does that mean they're closely related? Not necessarily. So to be homologous, it means it has to come from a common ancestor. And yes, birds and pterosaurs do have wings, but they're actually totally different from one another. Birds have a lovely type of wing pattern that's very different than what pterosaurs have. So this isn't the real tree at all. And in fact, for those of you who have ever owned a pet bird, you probably already know this, but you owned a dinosaur, right? Their attitude makes it very clear. So birds and dinosaurs actually have a bunch of traits in common, like an S-shaped neck, a fused clavicle, otherwise known as a wishbone. They have an ankle pulled off the ground, which is why they walk on their tiptoes. There's all these different things that unite them as a group. That means that wings evolved twice. And we actually know wings evolved more than that because we have bats too, right? We also have insects that have wings. So wings are something we're going to call homoplasy, not homology, but homoplasy, meaning evolving more than once. And I promise I'll keep using these words and explaining them so you don't have to remember all of these words. So Echinoderm homologies, meaning what they have in common, it's not as exciting as wings on dinosaurs, so we're not going to go into them too much, but I've color-coded all of these organisms so you can see the homologous parts. So we see that these red plates here, which I'm pointing to, in case you cannot see that red. So these are the oral plates. These blue plates here, these are the cover plates to the mouth. So all of these are homologous, and we're going to use these to make our big phylogenetic trees. All right, so these data sets, as I said, are constructed both by looking at museum collections that were collected over the past couple hundred years and by looking at field work. So here we have a photo of me here hanging out in the Czech Republic at the Prague National Museum looking at fossils that were collected in the 1800s with their original labels. It's really cool. And then I'm doing fieldwork in Sardinia, Italy. And here's one in Indiana. Maybe a little less exciting, but still fun. All right, so today we're going to be talking about a group of echinoderms called Blastozoans. Don't need to know anything about them, but the only thing I'm going to ask you to remember is that they have unique respiratory structures. So what I mean by that is if you take a look at this critter, you'll see that there's a bunch of holes punched all over the body. That's how it's breathing, right? So oxygenated seawater is coming kind of in through one, and then the deoxygenated seawater is going out through the other of those two little holes. So each blastozoan group has a special type of respiratory structure like that. So that's how we define them, right? All diploporans, meaning things with these little holes, have diplopores. Okay, so the problem, their morphologies, their shapes are really different. And we think that there's a lot of homoplasy, kind of like how birds and pterosaurs have wings, but they're not closely related. It's the same concept. So as I said, all diploporans have diplopores, which are a double pore respiratory structure, kind of like how you have lungs. These critters have diplopores. There's two little holes, dipore. That's where that word is coming from. All diploporans have diplopores, right? Okay, but some of them look a little different. All right, so these have a little bit different of a thing going on. They've got a bunch of canals connecting them, and they're constructed totally differently, but they're still diplopores. All right, and I lied to you. Some diploporans don't actually have diplopores at all. Some have no respiratory structures. We don't know how they breathe yet. We're still working on that one. And finally, we've started finding some echinoderms that are totally different. This is an Idrio asteroid, which you can envision as like a starfish slapped on top of a cookie. All right, that's kind of what they look like. And this has diplopores too. So what is going on? There's a couple ideas. One, either I have just sold you a whole bunch of nonsense, right? That's not a great way to start a talk. So that's hopefully not it. Or two, something more complex, something more complicated is happening with understanding these evolutionary relationships. So hopefully we're going to be able to use evolutionary theory to solve this. All right, so let's look at this. And it is the end of January. February 12th is Darwin Day. That's actually Charles Darwin's birthday. So happy early birthday, Charles Darwin. We're going to be using his theory of natural selection, of evolution by natural selection, to talk about this today. So this is the first known evolutionary tree. And Darwin scribbled this in his notes to say, I think that if all of these creatures here, we'll call them, these letters here, are related, that means they must have come from one group. And that's the theory that we'll be using today to kind of help us understand things going on. This is an evolutionary tree. I know, it does not look really pretty, so let's take a couple seconds to familiarize ourselves. Remember, this is just like a family tree. Here are all the descendants. This is where all the ancestors are. I've color-coded the groups, all right? So red is one group, yellow's another group, blue's another group, and then green is another group, okay? There are 29 species. There are 70 characters that we used, like round mouth or oval mouth or diplopores or no diplopores. That's how those characters are made. And this tree was constructed by using the fewest number of changes, which is something in science that we call parsimony. You might know this concept as Occam's razor, or if you hear hoofbeats, think horses, not zebras. That's exactly what parsimony is. It's the same thing. All right, so this is what that tree looks like. And what this tree is it's polyphyletic. And remember, we talked about that. That means that hippos and elephants were kind of shoved together into the same group, or birds and pterosaurs with wings, if we shove them together in the same group. It means that a whole bunch of different things have accidentally been shoved into the same container, which isn't a real evolutionary group. That gives us a problem, right? Because we've been assuming that they've been one group the whole time. So one thing that I want to note here is that there are 8.69 times 10 to the 36 possible combinations of this tree. This is one of the best ones. This is one of the ones that we use to say the fewest number of changes happened. But there's so many possibilities that I think that's just a wild number to look at. So for those of you who are just getting into phylogenetics, do not do this on a home computer. That's what happened to my grad school laptop, which I am no longer using. I'll say that much. So let's take a look at the different groups shown here. I've color-coded all of these, as I said, but all the red is showing up in one little area. Okay, that's a good thing. That means that they're all related to one another. There's nothing weird going on. These are called the spheronidids. All you need to know about them is that they're absolutely beautiful. They have short little arms. that are coming right off the body like this. We have the yellow group, Glyptosphaeratida. There will not be a pronunciation test at the end of this or a spelling test. So these have beautiful arms that dangle down the body. They kind of look like Christmas lights, if you can imagine with me kind of dangling down. But as you can see, all the yellow stuff isn't in one area. So it actually looks like there's more than one group here that we've been mistakenly calling one group. We have the astroblastids, which are really cool. I'll talk about those in a second. But all these green ones are non-Diplopores at all. These are other groups of echinoderms that we've put in here to see how they are related to other echinoderms. And what we can see is that we've got a whole bunch of weird critters showing up in the middle of our tree. So Diplopores are kind of a mess. Here are astroblastids. They look really cool. They're a group that shows up only in Russia for about 20 million years. We really don't understand them at all. So come back to me in about 10 years and maybe I'll have an answer for you. What does this mean and why do we care? Both excellent questions. So Diplopores, those respiratory structures that we talked about, are homoplastic, just like bird wings and pterosaur wings. They're re-evolving over and over and over through time. That means that we can't use them to define our evolutionary group. I can't say that all Diplopores have Diplopores. All right? Why? Does it mean that Diplopores are more helpful in oceans where certain things are happening, like maybe the water's a little bit warmer, so Diplopores are a little bit easier to breathe with? We don't yet know. But what we do know is that Diplopores have re-evolved a whole bunch of times throughout the evolutionary tree of echinoderms. And that is going to bring us to the next part of our talk. How can we use this to understand evolutionary trends through time? So I'm going to go back to this slide. Here's the tree we're looking at. This next slide looks a lot different, but it's the exact same tree. The only difference is it's now been plotted against geologic time. So these words, the Ordovician and Silurian, that's just geologic time. Here is the age in millions of years. All right. I'm going to add one more thing to this tree. And what I'm going to add is where these fossils were found. All right. So the geographic location of them. So anything with a square here, that's Baltica. Baltica is anywhere like Norway or Sweden and Estonia today. Gondwana is that large continent we were looking at that's like South America, Africa, Australia, Antarctica. And then we've got a whole bunch that were found in the United States, like the Southern Appalachian Basin, so similar to where North Carolina and Tennessee are today. We've got the Cincinnati Basin, where Ohio and parts of Kentucky are. So that's where these fossils were found. And what we're going to do is we're going to find out how these fossils moved evolutionarily and geographically at the same time. So if I know where the ancestors came from, and I can see where the descendants came from, I can start to plot out their specific pathways across Earth hundreds of millions of years ago, which is really cool, right? So again, to remind you, this is the Earth 480 million years ago. Here is Laurentia, where we are today. Here is that giant Gondwanan supercontinent that makes up Africa, Australia, Antarctica, and South America. And here we have a bunch of other little pieces of land. So echinoderms, as we said, were moving across time and space, but we really weren't sure because we didn't have that evolutionary tree to look at. Now we do. So this chart, I'm going to explain it really quickly. Again, here is North America, where we are. Here is Gondwana, that big, big, big continent. What this is showing is this is how echinoderms were moving. They were moving from Laurentia to Gondwana, and some were moving even further back. So why is this happening? Well, we think that echinoderms were using really warm circulation patterns, so warmer water that comes flowing through the ocean in certain patterns called gyres, right? So the water is circulating. They were kind of using it to catch a ride to be able to go to these different places. Now, echinoderms all have what we call a larval stage. So even the starfish today has a larval stage, a free-swimming larvae. Even the echinoderms that have big stems, they all have that free-swimming stage. So over millions of years, these larvae were actually moving further and further and further from where they evolved from to new places. And with this evolutionary tree and with their geographic locations, we can start to plot out how that actually happens. So now the next question is we know how they're related. We know how they moved. Can we start to answer why they moved? What caused them to pick up their bags and move to a new continent? Let's find out. All right, so this graph looks horrifying, right? So I'm going to replace it really fast with something else. Okay, so what I'm showing you here is what these variables are actually tracking. So carbon-13, as we said earlier, is tracking the amount of photosynthesis, more or less photosynthesis. Oxygen-18 is tracking the amount of sea ice. The more sea ice there is, the colder it is. Mercury over total organic carbon is just tracking the amount of volcanic activity. More total organic carbon over mercury is tracking more volcanoes going off. And this last one is tracking when our new species appeared geologic time-wise. So here is our geologic time. This is when new species appeared. So what are we doing? Can we see if there's specific events here that might be triggering more species to show up? We actually don't see that yet. We're still trying to figure out what's going on. And the reason that we still can't figure it out is that we're still trying to figure out globally what's happening. Most of these echinoderms do come from North America. So the more fossils that we find all over the world, the more we'll be able to fill this in. And the more we might be able to see how these echinoderms are responding. One of the only traits that we can really see is right here. We see that with all the volcanic activity here, we don't see new species appearing. So maybe echinoderms aren't huge fans of volcanic explosions. That would make sense, right? So we're still working on trying to figure that out. So the big question now that you might be sitting here thinking at home is, what can this tell us 440 million years later? Why does this matter, right? And one, it's really cool to learn about what the Earth was like in the past, but can this actually help our day-to-day lives today? And I think it can, and here's why. Why would echinoderms respond differently to climate change in the past than today? So today, starfish are evolving different patterns. They are getting sick from all of the higher acidity in the seawater and the higher temperatures. Starfish are responding to climate change happening today, which we know is happening at a rate faster than it's ever happened before in geologic history. So if echinoderms responded a certain way 440 million years ago, do you think that they would be responding differently? Probably not. They're probably responding in very similar ways. So if scientists can figure out exactly how echinoderms were moving, or the types of features that were more beneficial, we might be able to make a roadmap to help us understand how echinoderms might respond in the oceans today to climate change. So that's why this work is still important today. So here is my summary real quick. Better global sampling, so fossils from all over the world, helps us get a better, accurate idea of how evolutionary patterns were occurring throughout the geologic timescale. It helps me find out who was related to whom, how these organisms were moving, and the different types of features they had. We can use evolutionary theory, so Charles Darwin's evolutionary theory, by looking at who is related to whom and making these big, pretty trees, helps us understand not just who's related, but we can use it to understand how, why, and when the critters in the fossil record are changing. And we can use fossils from the past to help us understand and make a roadmap for how things might be responding to climate change in the future. It can help us kind of create a plan to save different creatures or how to preserve different environments, which can be really helpful. All right, and with that, I would love to thank all the folks that helped me with this work, that work with me, my collaborators, and I would love to thank my funding sources, which are all right here. Thank you so much for listening. I'm going to stop screen sharing so that I can see all of the questions really quick. All right, so a couple of questions have started coming in, and I will go ahead and answer them if everybody can still hear me. The first question that's come in is, why did I want to become a paleontologist? Which is a really fun question. So I actually started college thinking that I was going to be a music teacher. I was a flute performance and music education major in UNC Chapel Hill. And it turns out I really hated staying in the practice rooms all day playing my flute. So I started taking classes that weren't in music, and I came across a paleontology class completely by accident. And the professor was just so engaged and he loved what he was doing so much that I decided I wanted to do it too. So I did change my major that day, and I'm really glad that I did. The second question is, why study echinoderms? So the first answer to that is, you just looked at all those fossils of echinoderms, right? And they are just beautiful, right? They are gorgeous fossils. So I like invertebrates better than vertebrates. So I work with invertebrate fossils. And the reason for that is I think that when you go out into the field and you can find these little shells that hold so much of history, and you can find them and start to do these types of analyses, it's just so much, it's really fun for me, right? It's really a lot of fun to learn more about the history of life on Earth that way. So I study echinoderms too, because there's so many mysteries with them still of why they evolved these weird features. So that's really why I study them, is there's so many puzzles that I want to learn the answers to, and hopefully I will. Where were the first echinoderms found? That is an excellent question. So the first echinoderms are found in the fossil record about 530 some odd million years ago. All right, and those fossils are found in Cambrian age rocks. That's the name of the time, Cambrian. So those rocks are really rare. It's so old, and the oceans were really, really low at that point on Earth's history, which means we have not too many ocean rocks. So the first of those fossils that we find are places like Spain. There's some in China. Those are the two areas that have a lot of those. There's a couple places in Canada too. But those are where the earliest echinoderm fossils we know of were found. It's a great question. So where do you want to go to look for echinoderms? That is such a good question. Okay, so this is going to take a little bit of a road trip if you're listening in from New Jersey, but there's a lot of echinoderms in Indiana and Illinois. There's a lot in Tennessee. There are some in Virginia. And the closest place to you probably is going to be New York. So New York and Buffalo has a lot of echinoderms. And sometimes these places are really easily accessible. For example, there is a Walmart in Kentucky where you can just walk right behind it, and there's a whole ton of echinoderm fossils just right there. Sometimes you have to go out into the field. It's a little not as easy to get to, but echinoderm fossils, or any fossils, can be found in a lot of different places. New Jersey itself has a lot of cool fossils too. Right. So what was the biggest echinoderm? So some of the biggest echinoderms, oh my gosh, are like, there's some starfish today that are just giant. Oh my goodness, they're like this big. So those are some of the biggest ones that I know of today. Echinoderms move through a way that's really different to us. They bring in water into their body and they move it around. So there's a lot of pressure changes, which is how they're moving. We're not like you and me, where we can just kind of move around, not a whole lot of thinking about it. We don't have to move fluids to different parts of our body. Because of that, they can't get very large. There's constraints on how big they can get. So that's a really good question. All right. So would the animals handle respiration and waste removal differently with climate change? That is, find new ways or chemicals to excrete. That is such a good question. So echinoderms are always living in the ocean, always. They don't have a way to filter out salt in their body like we do with our kidneys. So they have to stay in the ocean water and salt water, or else they can't survive. So with climate change, they're probably doing some things differently because excuse me, because with the higher acidity in the water and the higher temperature changing, oxygen is diffused, pass through water a little bit differently. All right. So there are a lot of different things that they might be doing. So depending on the water currents, if there's more water current, they might actually evolve a slightly larger anus to help them handle waste a little bit differently, and different things like that. That's a really good question. So someone had a good comment here. You said all vertebrates had one bone, two bone, many bones in their limbs. Sharks are vertebrates. They do not have these bones and never had them. I apologize. I did mean to say tetrapods. So things that evolved from the first fish that walked on land. So tetrapods, things that have the 4 limbs on land today, which does include whales and dolphins. My apologies. Thank you for that correction. All right. So did diplopores help the creatures remove waste? Mary, that's a really great question. They didn't help them remove waste. So diplopores are only for respiration. So they're only using it to breathe. So kind of like how you breathe in oxygen and you breathe out carbon dioxide, they're working in the similar pattern, just with a very different structure to help them. All right. So they removed waste by having a separate hole for waste to excrete through. Echinoderms are actually really closely related to humans in that way, where they have a mouth and an anus that are separate. So most invertebrates don't have that, actually. So echinoderms are actually more closely related to vertebrates in that way. So another question is, I'm 12. Is there a good book to read? And there are a lot of really great books out there to read. So for 12, I'm not totally sure off the top of my head, but there's some really great books if you are interested in paleontology. One thing that you can go to is a website called www.timescavengers.org, and it's one word, timescavengers.org. And there's a whole list of different books and educational materials that you can go find for people who are interested in paleontology. It's a really great question. So are there any endangered echinoderms today? Yes, many echinoderms are endangered. They are not handling climate change extremely well. Echinoderms can be a little picky about the amount of sediment in their water supply, about the amount of acid in their water supply, and about the temperature in general. So there are many that are struggling a bit today. Many echinoderms also live near coral reefs, which many of us know are not doing so well. So there's a lot of things that we can do to protect our coral reefs. There's so much that we can do. So some things include using coral reef safe sunblock. There's types of sunblock you can buy that are safe for coral reefs. A lot of the kinds that you can get at CVS and other pharmacies aren't as safe. So we have to make sure to be careful with that, and things like not polluting, recycling, making sure our trash goes in the right place. That can actually really make a difference for a lot of our friends in the oceans. So another really great question from Sarah is ocean acidity uniform throughout the Earth's oceans and ocean temperature? So it's not. And I'm going to answer your question both as an answer of today and one through geologic time. So ocean acidity is not uniform throughout Earth's oceans today. The Pacific Ocean is slightly more acidic than the Atlantic Ocean. And the reason that has to deal with the water temperature and the water pressure. So the further down we go in the ocean column, the higher the pressure gets. Water under higher pressure can actually, it can hold more acidity into it, essentially. All right. So typically the further down in the water column we go, the water also gets colder. And cold water can also hold more gas as well, so CO2. So the colder and the more pressure we have with our water, the more acidic it is. That means that the Pacific Ocean, which is deeper than the Atlantic, will always have a little bit more acidity to it, and it will also have some more colder water. That means water at the North and South Pole in the deeper water is also extremely acidic as well. Throughout geologic time, our temperature on Earth does change. It gets warmer and colder. These are climate change cycles. That's not the reason why climate change is happening today. It's happening at a rate much faster than we've ever seen before. So it's not the same causes of it. The causes of climate change today are anthropogenic, meaning human caused. But we do see it getting warmer and colder, ice houses and hot houses. So ocean acidity does change over geologic time as well. That's such a good question. Thank you. All right, so a couple more questions that I saw coming through. What is the biggest question I am trying to solve right now? Okay, so the question I am most excited about solving right now is why those respiratory structures, the diplopores, those two little holes, why were they showing up so many times? So the question I'm trying to answer is it possible that in shallower water, they were really easy for oxygen to come in and out of the body, right? Or was it easier if you were living in an environment that had lots of sediment? So some diplopores were actually buried under the body plates, meaning there was, which could be, let me rephrase that question, which could be a response to like a whole bunch of sediment hitting it. And as you can imagine, if your respiratory structures are right there, you don't want sediment smacking into it all the time, right? In like shallow water in a coral reef environment. So maybe if we bury those diplopores, maybe that's why they're working the way they are. Maybe that's why they're helpful. We don't know yet. And that's one of some of the questions that I'm working on today. And I'm really excited about it. All right. And let me see, are there any other questions? Have any gone extinct recently? Some echinoderms, yes, have gone extinct recently. The echinoderms I look at, the ones that are really, really, really old, all went extinct hundreds of millions of years ago. There were a couple different mass extinction events. Many of us have learned a lot about the one that affected the non-bird dinosaurs. So the Cretaceous Paleogene extinction, the big asteroid that came in, we see a lot of that in like popular science news. That is the one that's really famous because that's where the big dinosaurs were taken out, not the birds. The birds are still here, right? If you don't believe me, go say hi to a Canadian goose. They're not so nice. They're dinosaurs, all right? No offense to Canadian geese. All right. So, but there's other extinction events that happen throughout time. So there was one called the Ordovician extinction event. There was one called the Devonian extinction event. And then there was the big one called the Permo-Triassic, which is also called very dramatically the Great Dying by scientists. So that one killed about 96% of all life on Earth, which is where most of my echinoderms, unfortunately, did not make it through. So the groups that we look at today are the only ones that survived that Permo-Triassic extinction. OHAV has an excellent question. Is it possible to correlate the amount of Diplopores in Diploporans with the amount of oxygen in ancient oceans? So essentially, OHAV is asking, if we have more respiratory structures, can we say there was more oxygen or less oxygen? I would hesitate to say yes, but I think it's possible that happens. I would say that the more diplopores, the more respiratory structures that an organism had, it might connect to less oxygen. And the reason would be, if there's less oxygen, you're going to have to work harder to get that oxygen into your body. The easiest way to do that is to get more and more and more respiratory structures. But that has a downside, because then you're using a lot of energy just trying to breathe. So we don't know for sure yet, but that is a good question, and it's possible that is happening. All right, so Catherine has found many pieces of crinoid stems in the Finger Lakes of upstate New York. Yes, those are echinoderms. So those crinoid stems are echinoderms in the Finger Lakes region of upstate New York. That's actually where my husband was born and raised. So I have gone up there and done some fossil collections. So that's been really fun. So the Finger Lakes area has some beautiful fossils. One thing that you can go do is go to Ithaca, where the PRI is, Paleontological Research Institution, has a lot of information on those crinoid stems. All right, so I think that might be all of the questions that I've seen come through. If one of the hosts has seen one that I have not.
All right, Sarah, thank you so much for your talk.
Thank you so much.
Sure, everyone's in the audience is thanking you as well, though we can't see them. And I really enjoyed that. Thank you for being with us. And thank you for taking so many questions.
Thank you all.
Our next talk will start at 11:30. So if you all just want to take a short break, we'll be back in about half an hour, a little more. Thank you.